This chapter is all about the cerebellum, which literally means little brain. And this is a neat looking picture, I think. You could see just the plain MRI on the left there. And then in the middle, you could see how the cerebellum is outlined. And then you can see the multicolored picture of the cerebellum on the right. So I think this is just kind of a neat looking slide to get us started. Basic function of our cerebellum is balance. Okay, so coordinated movement, balance, and motor learning, so muscle memory. And this is a really great example of when all of these things are very well practiced and everything is going very right. So Simone Biles has this coordinated movement, balance, and motor learning down to an elite athlete status here. So check out her balance on the balance beam and the focus there. So that takes a lot of muscle memory, a lot of coordination, and a lot of balance. So coordinates movement, it helps smooth it out. It's the quality control and correction. It detects problems and makes corrections. Balance and equilibrium, pathways for postural control and movement, locomotion. Motor learning, what muscle are we going to use? What amount of strength of contraction do we want to use? We don't want to overpower or underpower or overshoot or undershoot, and the velocity of it. The cerebellum is derived from the metencephalon. So remember chapter 2, tel, di, mes, met, mai. So this is metencephalon. You'll find it behind the pons and the medulla, posterior to that fourth ventricle, and it's going to sit on top of the foramen magnum. It sits in the posterior cranial fossa, and then the tent of the Tentorium Cerebelli is one of the separating fascial or connective tissue layers. So what we can see in this picture here, I'll take my red marker, so see the Tentorium Cerebelli up here as well. You can see the falx right here, and then the falx cerebri here. So making sure that you're looking for the word cerebri versus cerebelli, they're so similar. So this tentorium is this plane right here, more of that horizontal plane. As opposed to our falx, if you look off to the right here, you can see it's denoting it is in this plane, so going vertically. You can see tentorium over here on the right picture. You can see thalamus is labeled here. Corpus callosum would be right here. Tentorium Cerebelli. One of the things that I like to think of here is the word tentorium, tent. It's the tent over the cerebellum. Another thing to watch out for is whenever you see cerebelli or cerebri, do a double take and make sure that you're actually seeing cerebelli and not cerebri. Those words are so similar that people get them confused sometimes. So the Tentorium Cerebelli separates the occipital lobe from the cerebellum. So I'm going to draw on here. You can see where I starred it in yellow for the Tentorium Cerebelli to draw your eye there, and then you can see where that layer would be here. A little bit lower, there we go. You could see it on this side here. So I'll erase that so you can see it without my highlighter there. Just wanted to call your attention to where it is. And then the occipital lobe, recall that that is posterior, so in green here, occipital lobe area here. And then when I think of occipital lobe, the first thing that pops into my mind is it's the visual cortex. So that's the thing I associate with that. If intracranial pressure increases, then the cerebellar tonsils can herniate and put pressure on the respiratory control center of the medulla. So this area here, I'll take purple, This area down here, you can't really see my marker. I'll go for yellow again. There we go. This area down here, obviously we want that to stay up in the skull and not herniate through that foramen magnum. At the end of this lecture, we'll look at some of the dysfunctions associated with the cerebellum, and this is one that's not uncommon that we might see in practice. I've seen more than I can count of these cerebellar tonsillar ectopia, or sometimes called an Arnold-Chiari malformation. And we'll talk about the implications of that at the end when we go through our dysfunctions and diseases. This view helps us to visualize the posterior cranial fossa. So imagine that the top of the head has been removed and we're looking down so you can see where the ears are in the picture to the right. I will note that in a red pen. So these are the ears here. This is the nose at the front, so that's the view that you're looking at here. So if you look to the left side here now, you can see where I starred the Tentorium Cerebelli as the that kind of fascial layer on the left, and then you can see how the sinuses kind of fit in with that as well. And then looking off to the right, I starred in blue to grab your attention to the cerebellum in the posterior cranial fossa. So you could see what that looks like. And then you can kind of look around there. You can see where the falx cerebri is in the front area, and you can see that anterior cranial fossa, the middle cranial fossa, and then looking there again to that Tentorium Cerebelli, and then how the cerebellum sits in that posterior cranial fossa. Another view of the Tentorium Cerebelli and a couple of other structures. So if this is of interest to you or you need a refresher, it is chapter 4 in your Nolte book. So we could see the Tentorium Cerebelli and the falx cerebelli here. So falx in between the two hemispheres of the cerebellum, and then the Tentorium Cerebelli is that tent that was over the cerebellum. The pictures here to the right of the cerebellum, you can see the actual dissection on the left and then the drawing on the right to help kind of clarify things. So looking here, we can see that there's a right and a left hemisphere to the cerebellum. That's going to correlate to the same side of the body. Okay, so the right hemisphere of the cerebellum coordinates to the right side of the body, and then it's going to cross over and coordinate to the left side of the cerebral cortex. So left cerebral cortex, right cerebellum, right side of the body, and vice versa. Right cerebrum, left cerebellum, left side of the body. Really useful stuff when we're adjusting people that have a deficit on one side or the other. So you can adjust and just kind of motion palpate the major joints of the body. Let's say they're having trouble with their left side, or if they have left side cerebellar issues, maybe they've had a stroke or they have MS lesions in the left side of the cerebellum. Just motion palpate and adjust the left side of the body, leave the right side of the body alone. Adjust the spine accordingly, and then do the extremities differently. So that left side of the body extremity is all throughout. And then you can give them some exercises that we'll talk about at the end as well. So the idea that the cerebellum is ipsilateral, the cerebrum is contralateral is what I want to make sure that you guys get here. You can also see the vermis. I'll take my marker here. We'll see more of this, but it's nice that it's kind of an upside down V shape there for vermis, right? So vermis. And then the folia. So these folia, if you look, we have a folia and an arbor vitae. So the arbor vitae, if you look up top here in the drawing picture, arbor vitae, this area here, and then in the dissection picture, this area here, the white matter in there. Okay, so the arbor vitae is this internal core of white matter tree of life is what that means. I think it's kind of cool that they call it the tree of life. And then with our tree theme here, we have foliage, right? Just like you would have foliage on a tree, right? That foliage. I'll erase that so you can see it without my drawings in there, my scribbles. So the folia are the folds of the cerebellar cortex that covers the surface. It increases the surface matter. So we're going to see millions of neurons here. So we have way more surface matter or surface area for what's going on in here. Remember, this is a really high functioning area. So high performance, high maintenance it's stuff going on in here. And then you can also see the mention of the peduncles here. If you look off to the right, we have a superior, a middle, and inferior peduncle. Peduncle, remember, is just the stalk, so it's talking about these three areas right here. I'll erase that so you can see that as well. Just wanted to call your attention to it. We'll look at the peduncles in greater depth a little bit later. Just wanted to introduce that concept to you. And if you look down to the right, bottom right cartoon picture here, check this out. It is an upside down homunculus man kind of concept, right? So our homunculus man of the cerebrum, we also have a homunculus man or somatotopy of the cerebellum. So you can see here is kind of a lateral aspect here, right? So lateral aspect, we have upper extremity, lower extremity, and then this guy here, he's upside down. So that's the head, and this is the feet up here. Okay, so if there is an issue, let's say one of our MS patients has a lesion right here, they're going to have troubles with their right foot. So we'll get into this in more detail as well, and we'll get into the different nuclei of the cerebellum. Just a little intro slide, let you know what's coming. We saw the right and left hemisphere, and we also have lobes associated with this as well. So we have an anterior lobe, a posterior lobe, and a flocculonodular lobe. Yes, flocculonodular. Okay, kind of an interesting word there. So flocculo, I actually looked it up, it means fluffy. Okay, so it's a fluffy nodular lobe. So let's look at our picture here. We could see the flocculonodular lobe in blue here, and then note it is over here. Okay, so see that area where I'm moving around in blue? I'm going to go ahead and erase it so you can see it now. There you go. So that's the flocculonodular lobe, that little guy right there. And then the green is the posterior lobe, and the yellow, so down here, is the anterior lobe. Up top, the anterior lobe is obviously in that kind of pinky orange color. Otherwise, the blue and the green are the same for up top there. Our anterior and posterior lobe are separated by the primary fissure. Our posterior lateral fissure separates the posterior lobe from the flocculus. If we see increased intracranial pressure, it can cause a downward motion of that cerebellar tonsil, and it can put pressure on the reticular formation in the medulla that's going to control breathing and blood pressure. Things to note of the anterior lobe is that it is also called what is called the paleocerebellum. So we're going to see neocerebellum and archicerebellum as well. So this is right in the middle. So neo means new, archi is like archaic, and then paleo is going to be in the middle. So anterior lobe, not the newest, not the oldest aspect evolutionarily of our cerebellum. It compares commands with actual movement and helps make corrections of our movements that way. Our spinocerebellar tracts are going to go here, so from the spine to the cerebellum. This is going to get input from our muscle spindles, so stretch, and our Golgi tendons and joint receptors, so GTOs, remember, for tension of the muscle. We're also going to see input coming in from our inferior and superior cerebellar peduncles, so those stalks coming in and holding on to that cerebellum and the relays that come through there, and we'll go into details when we get into that aspect of the chapter. Posterior lobe, here's our neocerebellum, so this is the newest part. Okay, so posterior is our newest part, so that green aspect of the lobe. It predicts the movement of others and adjusts to the world moving around us. So posterior lobe predicts the movements of others and adjusts to the world moving around us. So when I think of this, I have the P for posterior lobe and the P for predicts, and then I carry this over to the grocery store called Publix. Okay, so I think of Publix and the fact that sometimes when we are shopping in these big stores, we think that maybe everybody else doesn't have this posterior lobe firing on all cylinders, right? So they're just kind of in their own world and not able to predict the movement of others and adjust to the world moving around them, and they slam right into you or don't even acknowledge that you are there. So that's my little trick to the mnemonic of what lobe is doing what. Posterior lobe predicts movement of others, and Publix. Okay, the grocery store. Cortex. So our cortex here is contralateral to the cortex and the corticopontine pathway and our middle cerebral, or I'm sorry, our cerebellar peduncle. So remember our cortex, it's going to the opposite side. So left side of the body, left side of the cerebellum, right cortex, and vice versa. So it's going to cross over and go up that corticopontine pathway. So pontine just tells us pons. Flocculonodular lobe. So this is the archicerebellum, so the oldest aspect. This has to do with basic balance. And we're going to see a connection with our vestibular nuclei. And it's connecting in via the inferior cerebellar peduncle. A quick recap of some of the basics here. So our phylogenetic classification, anatomical and functional. So archicerebellum is going to be the flocculonodular lobe, and it's vestibulocerebellum, maintenance of equilibrium. Paleocerebellum goes with the anterior lobe of the cerebellum, portions of the vermis, medial portion of the posterior lobe, and it's going to be Spinocerebellum and its regulation of muscle tone. Neocerebellum is going to be lateral portions of the posterior lobe, goes along with the pontocerebellum term, and that's for skilled movements. So the cerebellum, which literally means little brain, receives input from sensory systems, the cerebral cortex and other sites, and participates in the planning and coordination of movement. Our cerebellar outputs arise in a series of deep nuclei, the fastigial, interposed, and dentate nuclei, which we're going to take a look at on the next slide. So the next slide is going to be a slice. So if this is the front and this is the back, and in red is the cerebellum, it's going to be a slice through here, okay? Or it would be along this way. So if this were the knife, it would be slicing down this way. Okay, so slicing through here. Deep within the cerebellar white matter are 4 pairs of nuclei that contain most of the efferent neurons of the cerebellum. We have a mnemonic here. So from lateral to medial, it's don't eat greasy foods. So we have dentate nucleus in pink. That's the don't, the D for don't, D for dentate. Emboliform nucleus in blue. Globose nuclei is also in blue. And fastigial nucleus is in green. That center strip there is green also. That is the vermis. So the cortical regions have been color-coded to match their corresponding nuclei. The dentate nucleus is the largest of the cerebellar nuclei and extends into the cerebellar hemispheres. The cerebellar nuclei receive projections from Purkinje cells in the cerebellar cortex, while efferent fibers of the cerebellum can be assigned rather easily to anatomical structures. This is not true of the afferent fibers. I put a blank one in here for you, so go ahead and pause this recording and go back and forth from the previous slide to this slide to be able to fill out this picture. I, in the past, have used this picture verbatim on your final exam, so make sure that you know how to label this slide, please. To get a little bit more specific now, the dentate is for dexterity. So I remember the D and the D. So upper extremity or hand, specifically dexterity. And it's going to relate to the contralateral or opposite side red nucleus. Remember, the red nucleus in the rubrospinal tract is going to be for flexion protection of the upper extremity. It's going to relate to the contralateral thalamus. Going to give distal limb control to the same side. So remember that cerebellum is the same side of the body, opposite side of the cerebral cortex is its connections. That rubrospinal tract is actually called the dentorubrospinal tract, but more often than not you'll just see it called the rubrospinal tract. So it's actually going to start at the dentate and then the red nucleus. But we'll look at that in a minute. Emboliform and globose are also called the interposed nucleus, and our fastidial nucleus is connected with the flocculonodular lobe. So fastidial, flocculonodular, so F and F. And also with the vermis. We're going to see reticular and vestibular nuclei here, which are going to help control posture and balance and help control eye movements through the MLF. A reminder slide from our tracks PowerPoint about the rubrospinal tract. So rubro, red nucleus, ruby red, remember? So it's going to come from the cerebellum, that dentate nucleus, and red nucleus in the tegmentum of the midbrain. It's going to cross immediately through the ventral tegmental decussation, or crossing at the crossing. Descends through the pons and the medulla, and synapses at the motor neurons in the anterior horn of the cervical region in order to control the upper extremity movements, specifically flexion protection. So it functions in fine motor control of muscle movement. And then another thing we want to remember about the rubrospinal tract is that we're going to see deficits here in a Benedict's stroke. Okay, so a Benedict's stroke is going to give unwanted movements of the upper limb. Okay, so Benedict's stroke, you're going to see cranial nerve 3 deficits, and you're going to see unwanted movements in the upper limb, as opposed to a Weber's stroke, which is going to be the cranial nerve 3 deficits, but paralysis of the upper limb. So here we can see the rubrospinal tract at play right here. So Simone Biles attempting to regain her balance with instinctual arm reflexes. And the cool thing here is she's even wearing a ruby red leotard here. So red nucleus, rubrospinal tract, and the dentate nucleus connections here in order to help her get her balance back. Let's look at the functional divisions of the cerebellum. So the next couple of slides, quite a few slides actually, are going to discuss the details here of the vermis, paravermis, and the different aspects of the lateral hemispheres. We're going to look at the deep cerebellar nuclei. So there's a mnemonic here for the deep cerebellar nuclei. Those are some weird looking words if you've never seen them before. The mnemonic is don't eat greasy foods. DEGF, don't eat greasy foods. and it's dentate, emboliform, globose, and fastigial. So let's go ahead and look at the details here for these structures. Here we can see some functional divisions of the cerebellum. So if you look at the picture on the bottom, you could see the Spinocerebellum. I color-coded it for you, so in pink, Spinocerebellum. And then that's going to have the anterior lobe, the vermis, and the perivermis. And then the Cerebrocerebellum is has a little blue dot next to it there for you to see. And that's going to include our posterior lobe and the lateral hemispheres. The Vestibulocerebellum is going to be the Flocculonodular Lobe. So the anterior and posterior lobe, if you look up to the right there, you can see that there is a homunculus somatotopy to them. So we're going to see the trunk, head, neck area, the main medial area of the body is going to be the medial area of the cerebellum, and then the extremities are going to be more lateral. So Spinocerebellum, which is this upper area, okay, so upper area, receives input from our receptors from the muscle spindles and GTOs, so stretch and tendon tension, and joint receptors of the body and the face. We're going to see input from spinocerebellar tracts and the olivocerebellar tracts, so from the olive to the cerebellum, short little trip there. Olives are in the medulla, remember? And we're going to see here the globose and emboliform nuclei sending a message out to the contralateral red nucleus. So it would be more of a globo-rubro or emboli-rubro pathway. And it crosses over and descends to the spinal cord, and we just call it the rubrospinal pathway after that. Cerebrocerebellum, so cerebral cortex to the cerebellum. So it's going to receive input from the contralateral cerebral cortex. So that's repeated a lot in this chapter. So it's important to remember that it is opposite cerebrum from the cerebellum, and then ipsilateral cerebellum to the same side of the body. It's really important for functional rehab to understand that. Pontocerebellum is just the pons to the cerebellum, so short little trip there as well. Dentate nucleus of the cerebellum sends a message out to the opposite red nucleus. We call it the dentorubro, and it crosses over and goes down to the spinal cord. We're calling it rubrospinal after that, remember. Dentate nucleus sends a message to the opposite thalamus. We call it the dentothalamic nucleus. And then it sends the message to the cortex with the message of its perfect motor plan. Dentate nucleus sends a message to contralateral red nucleus, then to the thalamus. We call it the dentorubro-thalamic pathway. So a lot of words coming at you there. The thing to remember is it's going to tell you, right? So dentate nucleus of the cerebellum. Rubro, red nucleus. Thalamic, thalamus. So we don't have to memorize all of these pathways. We just have to know the nomenclature. We're like, all right, so it's going here and then here, and what are these things doing? Vestibulocerebellum receives input from the same side, ipsilateral inner ear and vestibular nuclei. And we're going to call them the vestibulocerebellar tracts, so the pathways. Vestigial nucleus is going to go to the ipsilateral or same side vestibular nucleus. We're going to call our vestibulospinal pathway once it's going to the spine. The MLF, or the medial longitudinal fasciculus, to stimulate 3, 4, and 6 to move the eyes. So remember this Yolk's vision. Then we can understand the extraocular muscles pathways a little bit here. This is a great picture to give us an idea of the cellular layer here. So we can see our molecular layer. I'm going to use my red highlighter here to kind of show you this layer. So our molecular layer is going to have our stellate cells, our basket cells. You can see a basket cell here, this little guy here. Basket cells are parallel fibers right here. You can see them nice and parallel to each other, parallel fibers of granule cells here. And then we're going to see our Purkinje layer with the Purkinje cells, nice and simple there, right? So you can see the Purkinje cells, these little purple droplet looking things here. And then our granule layer, I'll use my blue marker, keeping in theme here. Granule layer, we're going to see the granule cells, these little dots, blue dots here are granule cells. And we're going to see Golgi cells in this layer as well. We don't see them on here, but that's okay. And then we'll see our mossy fibers here. So I'll highlight that for you. Coming up, this green one. And then we'll see our climbing fibers here. So climbing all the way up. So this is really going to give us the control that we're looking for what we call neural sharpening. So we want to make sure that the most important stimulus is taken care of at that time. So it's not over or undershooting, and it's just the right amount, and this is how that's all orchestrated. I put this blank slide in here for you so you could practice writing the words. I find that to be the easiest way to study it. Kind of flip back and forth and fill that out so you can start to acclimate yourself to the names of these structures. So we can see our picture here to the right and the table of the names and definitions of these different cell types. So our climbing fibers, the definition is that they're axons of neurons in the inferior olive, so think medulla, and its associated nuclei. Mossy fibers, axons of neurons in the pontine nuclei and spinal cord and vestibular nuclei. So our pontocerebellar, spinocerebellar, and vestibular tracts. Our parallel fibers are axons of granule cells, and our granule cells are the interneurons of the cerebellar cortex. Our Purkinje cells are the only efferent cells of the cerebellar cortex and exert an inhibitory effect. There is one climbing fiber for one Purkinje fiber. Let's take a look at this diagram of the synaptic circuitry of the cerebellum. The cerebellum comprises 10% of the mass of the brain, but contains 50% of its neurons. This enormous population, for example, the cerebellar granule cells alone may number in excess of 100 billion. So this huge population is composed of a few cell types arranged in a repetitive, highly ordered array. This repetition of simple elements has led to the description of the cerebellum as an intricate synaptic computer for motor coordination. The basic cerebellar circuitry involves afferents, including mossy and climbing fibers. Climbing fibers originate from the inferior olivary complex and form multiple excitatory synapses on the cell bodies and proximal dendritic tree of the Purkinje cells. Collateral branches synapse in the deep cerebellar nuclei. Mossy fibers originate in the vestibular and pontine nuclei and the spinal cord to form excitatory contacts with granule cells in synaptic complexes called cerebellar glomeruli. Some branches excite local inhibitory neurons, and collaterals also enter the cerebellar nuclei. The axons of granule cells form parallel fibers that establish excitatory synapses on the dendritic trees of Purkinje cells. The Purkinje cells in turn send their axons mostly to the cerebellar nuclei. Some are also going to go to the vestibular nuclei. So at that cerebellar nuclei is where they're going to make inhibitory synapses. Some neurotransmitters in this pathway have been established. So local inhibitory neurons and Purkinje cells themselves use GABA. GABA is our inhibitory neurotransmitter, while granule cells employ glutamate. Glutamate is an excitatory neurotransmitter. Glutamate is probably also involved at the mossy and climbing fiber synapses. The principal cerebellar efferent axons arise from the cerebellar nuclei. The circuitry combines direct activation, so our afferents to granule cells to Purkinje cells, and indirect inhibition, so afferents to inhibitory interneurons to the Purkinje cells, which may be integrated in a complex spatial pattern and temporal sequence in the cerebellar cortex and deep nuclei to provide indirect feedback control for motor coordination. This is a good slide to study from now that we have had the introduction to the different terminology here. So if we start with the top left here, spinal inputs, we can see this Spinocerebellum aspect of the cerebellum that we looked at, the pontine inputs or the pontocerebellum, and then vestibular inputs or vestibulocerebellum. And then looking over here, it's the don't eat greasy foods or dentate, emboliform, globose, and fastidial nuclei. And if you look over here, we can see our lateral vestibular nucleus is the medial descending systems, and it's for execution of movement. Emboliform and globose, lateral descending systems, also execution of movement. Dentate nucleus, premotor cortex, planning and programming. Vestibular nuclei for balance and oculomotor function or eye movement. And this is to help you out if you like to study this way. Go ahead and pause here and fill these in. I find that to be a useful way to help to understand it. The next series of slides are really great for understanding structure. They come from your theme book, which is a recommended book. And let's just go right on through it. So what are we looking at here? We can see the tonsils here, and this would go into the foramen magnum if they had cerebellar tonsillar ectopia or Arnold-Chiari malformation. So hopefully these are right above the foramen magnum, just to orient you on where we are in the cerebellum here. I'm going to erase this so it doesn't get too busy looking. So we can see the tonsils, and then note this vallecula kind of looks like the word valley, right? And so we see this valley for the vallecula, and then another V word coming right on up here, so you can kind of see this as a V. That is the vermis and the pyramids of the vermis, and then the central lobule. And then when we look here, we could see these cut areas are the peduncles. Okay, so this whole area here is the cut peduncle being stalk, and we can see the superior cerebellar peduncle. Middle is always going to be the biggest one, okay? Middle cerebellar peduncle, and then inferior cerebellar peduncle. We can see the horizontal fissure, this right here, horizontal fissure. What else do we see here? We can see the uvula of the vermis, the little hang down of the back of the throat, right? We call that the uvula. We have a uvula here at the vermis as well. Moving over to the right, you can see the flocculonodular lobe. So you can see the nodule area, and then the flocculus. I'll switch colors here so you can see it. I'll go to blue. This is that flocculus area here. It means fluffy. Okay, so somebody thought that looked fluffy and called it the fluffy node there, fluffy lobe. And then I'll switch to red, and you can see where the fourth ventricle is here. I'm going to erase some of these now so you can take a look at that. There you go. And then the next slide is blank. I highly recommend you flip back and forth and study that way by now filling in the blanks. So pause and take as much time as needed. This is a lateral view of the cerebellum, the pons, medulla, some midbrain there up at the top as well, and cerebral peduncle that we can see. So we're looking at a side view, and I want to point out we've got medulla here, and we've got our green olives of the medulla. Right here would be the decussation of the pyramids. This would be the pyramid here. You can kind of see the other pyramid here just because it's a different kind of rotated angle that we're looking at here. Coming up superior from the medulla, we have the pons here. Coming superior from the pons, we can see the midbrain area. So medulla, pons, midbrain. Specifically, these are the cerebral peduncles, or where the cerebrum will be attached to. So peduncle just means stalk, remember. And then, of course, we have our cerebellum here. Note the little flocculus guy is hanging out right in there, little fluffy guy right there. And then our anterior lobe here in yellow. Coming to the primary fissure right here. Primary fissure separates the anterior lobe of the cerebellum from the posterior lobe. Do not get fooled by this horizontal fissure right here. Horizontal fissure is posterior and posterior lobe. Okay? So this is all just posterior lobe here, all the way to here. The little blue guy is flocculonodular. Up top in yellow is anterior and red is posterior. So posterior lateral fissure is going to separate the posterior lobe of the cerebellum from the flocculonodular. Okay, so posterior lateral fissure. And a nice clean slide for you here to practice. Here's a left lateral view of the cerebellum and lateral portions of the pons removed. This dissection, which has been prepared here to show the fiber structure, clearly shows the course of these cerebellar tracts. So let's take a look here. You can see anterior spinocerebellar tracts right here, spinocerebellar. Here with my blue marker is the middle cerebellar peduncle. That's the biggest of our cerebellar peduncles. We can see, I'll highlight it in green here, the superior cerebellar peduncle, this right through here. And then in yellow, I'll go for the inferior cerebellar peduncle coming up this way. This is a little too far up. Okay, right in here. And then you can see the olives of the medulla right here, one of the olives. This guy right here. What else do we see? We see the vestibular cochlear nerve right here. We can see facial nerve right next to him, right there. A big old trigeminal nerve right here. The size of the cerebellar peduncles, and thus the mass entering and emerging axons, is substantial and reflects the extensive neural connections in the cerebellum. The cerebellum requires these numerous connections because it is an integrating center for coordination of fine movements, balance. In particular, it contains and processes vestibular and proprioceptive afferents, so balance and where are we in space. And it modulates motor nuclei in other brain regions and in the spinal cord as well. And here is your practice slide. So go ahead and pause and go back and forth and make sure that you can identify these structures. This is just another perspective on the peduncles. So you could see the middle cerebellar peduncle, again the largest of them here. You could see the inferior here, superior up here. We can see the little trochlear nerve up here, cranial nerve 4, only one coming from the posterior aspect, remember. And then we see the colliculi here, the corpora quadrigemina. So up here would be superior colliculus for visual reflex, and down here is the inferior colliculus for auditory reflex. So superior is you see something out of the corner of your eye, turn and look. Inferior is you hear something out of the ordinary, turn and look. And what else do we see? We could see trigeminal nerve here, vestibular cochlear, facial, nervus intermedius, facial's buddy there, and then glossopharyngeals here, right there. And then we could see some olives here again. Oh, this guy, this little bumpy guy here. Great. And here's your practice slide. So go ahead and pause and make sure that you can identify these structures. Another perspective for us here, up here are the bilateral nuclei of the thalami. We've got our superior and inferior colliculi, superior, inferior. There's that trochlear nerve, just so you can kind of orient yourself on where you are. Cerebellum has been cut away from over here. The red is the superior cerebellar peduncle, blue is middle, yellow is inferior. Superior and inferior are both afferent and efferent. Middle is solely afferent. A couple of cool pictures, I think, here. So I got these from the human connectome. These are the functional MRIs showing the peduncles. Superior cerebellar peduncles, also known as brachia conjunctiva. So they emerge from the upper and medial part of the white matter of the hemispheres and are placed under cover of the upper part of the cerebellum. They're joined by the anterior medullary velum and can be followed upward as far as the inferior colliculi, under which they disappear. Below, they form the upper lateral boundaries of the fourth ventricle, but as they ascend, they converge on the dorsal aspect of the ventricle and assist in roofing it in. The ventral spinocerebellar tract enters the cerebellum through the superior cerebellar peduncles, which otherwise mostly contain efferent fibers. So we have our ventral spinocerebellar, and then we have efferent fibers. Superior cerebellar peduncle afferents are the ventral spinocerebellar, so sensory fibers from below L2, small portion of rostral spinocerebellar, and then tectocerebellar. So tectal, again, being tectum or the corpora quadrigemina, or the superior colliculus and inferior colliculus, all part of the same thing there. So that's the visual and auditory stimulus being able to respond to that stimulus. Efferents are motor pathways. Dentorubro pathway, so dentate nucleus to the contralateral red nucleus. So that rubrospinal pathway, so contralateral side to the flexor muscles of the upper extremity especially. Dentothalamic, dentate nucleus to opposite thalamus to cortex. This is going to give us our perfect motor plan. Dentorubrothalamic pathway, so the dentate nucleus to the opposite red nucleus to the thalamus to the cortex. Interposed rubro pathway, so contralateral red nucleus and the rubrospinal pathway, and it is our contralateral flexor muscles of the upper limb. That dentatorubothalamic cortical pathway, that's quite a mouthful. Let's kind of break that down a little bit here with this slide, shall we? So it's an efferent from the cerebellum, and via this pathway, activity in the dentate nucleus of the cerebellum and other deep cerebellar nuclei modulates activity in the contralateral motor cortex. So this crossed connection to the contralateral motor cortex helps to explain why each cerebellar hemisphere regulates coordination and muscle tone on the same side of the body. Middle cerebellar peduncles, as you can see in yellow there, that's the human connectome functional MRI. Afferent fibers here, so it's pontocerebellar. So ponto just telling us it's the pons, so cortico-pontocerebellar fibers. Gives us a motor plan from the cortex to the pontine nuclei. Decussates through the middle cerebellar peduncle. So afferent, remember, usually we associate this with sensory, but it just means that it's coming into this this area, into whatever structure we're talking about. The middle cerebellar peduncles are paired structures. So we have a left and a right, just like all of our peduncles. And these connect the cerebellum to the pons and are composed entirely of centripetal fibers, which arise from the cells of the nuclei pontus of the opposite side and end at the cerebellar cortex. The fibers are arranged in 3 fasciculi, so a superior, an inferior, and deep. Inferior cerebellar peduncle, the upper part of the posterior district of the medulla oblongata is occupied by the inferior cerebellar peduncle. It's a thick, rope-like strand situated between the lower part of the fourth ventricle and the roots of the glossopharyngeal and vagus nerves. Each cerebellar inferior peduncle connects the spinal cord and medulla oblongata with the cerebellum and comprises the juxta-restiform body and restiform body. Important fibers running through the inferior cerebellar peduncle include the spinocerebellar tract and axons from the inferior olivary nucleus of the medulla, among others. Inferior cerebellar peduncle afferents are going to be the dorsal spinocerebellar tract coming from C8 to L2, so that region of C8 to L2. Cuneo-cerebellar tract, rostrospinocerebellar tract, vestibulocerebellar tract, so the inner ear, olivocerebellar tract, or the contralateral inferior olives, so the climbing fibers, reticular cerebellar tract from the reticular formation. Efferents are going to be cerebello-reticular, so that's coming from the fastigial nucleus. Cerebellar vestibular from the fastigiobulbar, and the vestibulospinal, or the MLF, to the eyes. Inferior cerebellar peduncle carries many types of input and output fibers that are mainly concerned with integrating proprioceptive sensory input with motor vestibular function, such as balance and posture maintenance. Proprioceptive information from the body is carried to the cerebellum via the posterior spinocerebellar tract, which passes through the inferior cerebellar peduncle and synapses within the spinocerebellum. Vestibular information projects onto the vestibulocerebellum, also known as the archicerebellum sometimes. This peduncle, inferior peduncle, also carries information directly from the Purkinje cells to the vestibular nuclei in the dorsal brainstem located at the junction between the pons and the medulla oblongata. A little chart here to try and help you kind of keep it organized. So superior, middle, and inferior peduncles here. So superior afferent is ventral, spinocerebellar, tectocerebellar, part of the rostrocerebellar. We see the efferents being dentorubro, dentothalamic, dentorubrothalamic, and interposed rubro pathways. Middle only has our afferents being pontocerebellar, corticopontocerebellar here. Inferior is the vestibulocerebellar, olivocerebellar, dorsal spinocerebellar, make sure you know that one, we see that in tracks. Cuneo-cerebellar, reticulocerebellar, and the rostrospinocerebellar. Efferents are going to be the cerebellar, reticular, cerebellar vestibular, being the vestigiobulbar. It's been a lot of information in this cerebellum chapter, so let's go through some of the main vocabulary that I want to make sure that you take away from this, because there was a lot of minutiae, fine detail. So make sure that you know the anterior lobe. It's anterior to the primary fissure, so partly vermis, partly hemisphere. Flocculus, the hemispherical part of the flocculonodular lobe, functions as the vestibular system and eye movements. Nodulus is the vermal part of the flocculonodular lobe. It functions as the vestibular system and eye movements. Hemispheres are the large paired lateral parts important for motor planning and coordination of the limbs. Posterior lobe of the cerebellum is posterior to the primary fissure, partly vermis, partly hemispheric, and it's between the anterior lobe and the flocculonodular lobe. Tonsils are a medial inferior part of the posterior lobe hemispheres, adjacent to the medulla as it passes through the foramen magnum, and this is what might herniate down into that foramen magnum, and we would call it a cerebellar tonsillar ectopia or an Arnold-Chiari malformation. Vermis is the most medial zone of the cerebellum, straddling the midline, and vermis is Latin for worm. Let's talk a little bit about what the deficits might be. So our classification being functional, phylogenetic, and anatomical, and then what the deficits might look like. So our vestibulocerebellum would be archicerebellum, flocculonodular lobe. Okay? The deficit that we would see if this flocculonodular lobe was damaged would be truncal stance and gait ataxia. Now, what's ataxia? See the word part taxi in there? Movement. We might see vertigo, nystagmus, that kind of wiggle waggle to the eye, and vomiting. So they're going to have that vertigo, and the vertigo would lead to vomiting. Functional classification of the Spinocerebellum would be the Paleocerebellum. That's the anterior lobe, some parts of the vermis, parts of the posterior lobe, being the medial parts of the posterior lobe. Deficits, if this anterior lobe, some vermis, some posterior aspects, or medial aspects of the posterior lobe, if there's damage there, we would see ataxia, chiefly affecting the lower limb. We would see oculomotor dysfunction, so eye movement dysfunctions, speech disorders, and then our functional classification of pontocerebellum goes with our neocerebellum. That's the posterior lobe and our hemispheres of the cerebellum. If there's a deficit here, we're going to see dysmetria and hypermetria. So we would see a positive rebound test, and we're gonna take a look at what these things mean in a few minutes here. I just wanted to give you kind of a definition and a glossary to kind of look at. We're gonna see intention tremor as opposed to resting tremor. We're gonna see nystagmus and a decreased muscle tone. So let's go ahead and take a look and see what all of this means. Some of the ways that we test the cerebellum and look for deficits would be to watch the patient's gait or how they walk. We're going to do heel to toe, basically like walking a tightrope. Single leg stance, as our yoga club here is demonstrating. And then heel to shin, as you'll see in your lab for this week. Romberg's test, rebound test, past pointing, and pinky to nose. These are all cerebellar testing that we will become familiar with in lab. Here is the sheet that I want you to work from. So I put together this table for you here to kind of figure out what's going on with the cerebellum. So rule of thumb with the cerebellum, and with anything really, we want to stimulate the weak side. Okay, so stimulate the weak cerebellum side and the contralateral cortex side. So if the right cerebellum is weak, the right side of the body is going to have a deficit, and the left cortex would also need to be stimulated as well. We want to strengthen that first, then equal therapies after balance has been achieved. So the sheet here that I have for you for your lab would be the head tilt, Romberg's single leg stance, gait, or just watch them walk. Have them do a heel to toe walk, heel walk and toe walk while they're at it, right? So L5 and S1, checking in on that as we're checking in on their balance. And then heel to shin, so sometimes they can't walk, or we're working with a patient that can't walk, and we can just have them scrape their heel down their shin, and that'll give us the information we're looking for as well. Our eyes, we're going to look for square waves, or these kind of jerks to the eyes, and this fixation and stability. We're going to look for eye movements in the cardinal fields of gaze, convergence and divergence eye movements. With a flashlight, we're going to look and see what the reaction to light looks like. Optokinetics or OPKs, saccades and anti-saccades, pinky to nose test, pass pointing, rebound and drift, and we're going to also look at the cranial nerves as part of this balance workup here. Cerebellar lesions may remain clinically silent for some time because other brain regions can functionally compensate for them with reasonable effectiveness. Exceptions are direct lesions of the efferent cerebellar nuclei, which cannot usually be clinically compensated for. So some cerebellar symptoms when our patients come in and they exhibit asynergy, so a difference from one side to the other. So yeah, I'm right-handed, right? So my right side should be a little bit more coordinated and a little bit stronger than my left side. But the rule of thumb for that would be a 10% difference. There really shouldn't be more of a 10% difference between the right and the left side. And then if my right side were weaker and less coordinated or equal to my left side, that would probably be a sign of a deficit as well. Ataxia, so not moving appropriately. Decreased muscle tone. Intention tremor, and we'll watch a video on what that looks like. Rebound phenomenon. Lack of coordination among different muscle groups, especially in the performance of fine movements. Uncoordinated sequence of movements. Truncal ataxia. Patient cannot sit quietly upright or calmly upright and is distinguished from stance and gait ataxia. So we see impaired limb movements and an unsteady gait, just like in inebriation. So someone that is drunk and trying to walk and talk and coordinate movement, that is the cerebellum that's being affected. So think of when you saw someone drunk and what that looked like. That's cerebellum. Ipsilateral muscle weakness and rapid fatigability. So the patient here stands with one leg spread apart and places his hand on the wall for stability in our picture here. And then we see an involuntary rhythmical wavering movement of the hand when a purposeful movement is attempted. That's our intention tremor. Okay? Or as in the finger to nose test. And then we see this test indicating a cerebellar lesion here. And then we can also see the patient with their eyes closed is told to move the arm against resistance from the examiner. And when the examiner suddenly releases the arm, it forcibly rebounds towards the patient, or what we call hypermetria. I'm not a fan of this one. Don't do it in front of their face and have them hit their face. That's, you know, it's a legit test, but it is not good for patient retention. Right? We're going to look here at midline dysfunction. So check out this video here. It's going to show you gait alterations, titubations, spontaneous nystagmus, and affective disturbance. If the cerebellum is damaged in the lateral area, we will see this lateral dysfunction as a jerky, irregular movement or ataxia. We'd see broad-based gait, hypotonia or lack of proper tone, dysarthria, which is a condition in which the muscles you use for speech are weak or you have difficulty controlling them. Dysarthria often is characterized by slurred or slow speech that can be difficult to understand. And I put a link at the bottom of the page here for Selma Blair, who was diagnosed with multiple sclerosis and gave an interview, very bravely gave an interview, even though she was suffering from dysarthria. So her pattern of speech is a good example of the dysarthria. So take some time and watch that, please. Dysmetria is a lack of coordination of movement typified by the undershoot or overshoot of an intended position with the hand, arm, leg, or eye. It's a type of ataxia and can include the inability to judge distance or scale. Dysdiacokinesis, or DDK for short, is the term for an impaired ability to perform rapid alternating movements. You're going to see this in your lab. So a lot of this is really just preparing you for the terminology that you're going to see in the lab video. Tremor is an involuntary, somewhat rhythmic muscle contraction and relaxation involved with oscillations or twitching movements. So an intention tremor is a salient feature of multiple sclerosis. A pill rolling or resting tremor is a salient feature of Parkinsonism, and that's more the basal nucleus. That's not the cerebellum. Cerebellum would be MS and the intention tremor. We'll see an impaired check and rebound, impaired spatial recognition, cognition, understanding how far a distance is, and then mood disturbances. So spinocerebellar ataxia will see a degeneration of the inferior olive of the medulla, pons or cerebellum, and we'll see gait and limb ataxia and dysarthria. This can be genetic. Friedreich's ataxia. So this is a degeneration of the peripheral nerves, spinocerebellar tracts, dorsal column, so that fasciculus cuneatus and gracilis, and the corticospinal tract, so motor. Begins in childhood or adolescence, and we see an ataxia of gait and limbs, scoliosis, limb weakness, loss of vibration, and proprioception. This is genetic. Sporadic olivopontocerebellar atrophy, not really a known cause, gradual loss of inferior olives, pons, and cerebellar cortex. We see an ataxia of the gait and limbs and dysarthria, truncal and limb tremors. Alcoholic cerebellar degeneration, so Wernicke-Korsakoff syndrome. So this is chronic alcoholism. We see an anterior and superior vermis problem, and we see ataxia of gait, but speech and upper extremity movements can still be intact. So clinical applications here. What do we want to do for a functional neurological exam for the cerebellum and the cortex? So we want to test and localize the site of the weakness. We want to stimulate the weak cerebellar side and contralateral cortex side. Strengthen that first, then equal therapies once it's balanced. The head is going to tilt toward the strong cerebellar side. We will sway towards the weak side. We want to do cortical stimulation therapy, so scent is a really good one. Scent to one side. So think about the olfactory nerve and it going straight in, and it's going to give a good cortical stimulation to one side. So you want to have a pulse ox on hand. Pulse ox on hand, or your finger. Okay. I didn't mean that, but it's a good one. Little neurology joke. So pulse ox on hand there to check and make sure you're not pushing past the metabolic rate during treatment. Your patient will start to crash, and you'll see the heart rate either go up or down, and you'll see the oxygen saturations go down. So if it goes down, I personally don't like to see it go below 96. So some people will push it a little bit further than that, but I want really good oxygenation. So I'm going to stop them at 96, have them take a couple of deep cleansing breaths, nice and relaxing, and then go again. So you just keep the pulse ox on them while you're doing the therapies and kind of check in. If you see other signs that they're starting to not do so well, I mean, don't just rely on the pulse ox, just take a break. What we want to do is we want to localize to the cerebellum and the cerebrum. So on our piece of paper, when we are checking out the strengths and the weaknesses, we just put little check marks next to, okay, that was a sign that the left cerebral cortex isn't working very well, or that's a sign that the right cerebellum isn't working very well. So we would just put little check marks here. Okay. And then whoever has the most check marks at the end is what you're going to work on. So you'd end up with, you know, something that looks kind of like that. And you're like, all right, right cerebellum, left cortex. Okay, so it wouldn't be necessarily nothing on either of these. Okay, so if we determine that, then we're going to work right cerebellum, left cortex, until we start to see it kind of equal out. And that's going to take some time. Days, weeks, months, not sure. It depends on the person and the deficit. Things to consider. So not just cerebellum, but things to consider while we're doing these. So what tracts and structures are you stimulating if you add these things in? So vibration to the lower extremity. Okay, so this is the stuff I want you to think about here. So what is going to give us what's responsible for vibration sense? What happens if we put temperature changes to the upper extremity? Ice, heat. Okay, what are we going to stimulate? What are we stimulating with eyes open? What are we stimulating with eyes closed? Scent in one nostril, an earbud in one ear. Cover one eye. Think about the visual pathway drawing and what that entails. How about vibration sense on a lower extremity plus one eye closed and one ear has an earbud in it, and then a scent swiped under one nostril. What's going on there? There's a lot of input. Okay, so obviously you don't do that to them on their first visit if they've got a lot of deficits. You would build up to treatment strategies like that. Okay, great. That is the lecture portion. Now would be a really good time to take a little break and then go to your lab portion of this class. So she's going to have not only the actual lab dissection specimens, but videos of deficits of actual patients as well. So it's really great lab. Please check it out and answer your quiz questions on the website, and email me if you have any questions, comments, or concerns. Thank you.